So I'm going to talk about one of the projects that Lance Barrett-Leonard and I are working on at the Coastal Ocean Research Institute in genetics, and everyone loves genetics after coffee, but I'll try not to throw too much in. It's like stats without stats, genetics without genetics. So most of you know a lot about the killer whales in BC and the populations. Everything really revolves around the family unit. For most of these animals, they spend all, if not large portion of their lives with their family. They cooperatively hunt and feed, and so their family is really everything. But when it comes to mating, the family can't be everything. It's pretty much a problem. Inbreeding is bad. As a general statement, you just can't do it. You end up with this buildup of deleterious mutations, which causes higher prevalence of diseases, and just, it's not good. It's not a healthy way to live. And so a lot of species have developed ways to kind of ensure this doesn't happen. And for killer whales, however, where they're spending so much time, how are they choosing mates outside of the social network when they don't have this cool technology? So we know for northern residents, one of the ways they get around this from past work that Lance did as part of his PhD is they are using some acoustic cues. So we've got 3 different acoustic clans of northern residents, A clan, G clan, and R clan, and they're actually choosing mates outside of their own acoustic clan. So possibly picking up on some acoustic signals as to, okay, you sound different than me, there's a good bet we're not related, so I can go mate with you. But we don't know what's happening when it comes to Bigg's killer whales or the transients. And so when you want to understand how whales are choosing their mates, we really have to step back and say, okay, well, who are they actually mating with? That's the first step. So I do a lot of paternity testing. And we do this with what's called microsatellites, these small kind of repetitive sequences of DNA where you get half from mom and half from dad, but who cares about genetics? Let's talk about shapes and colors. So we're going to talk about each kind of microsatellite or piece of DNA is going to be represented by a different shape, and the variations of these shapes, variations of these kind of sequences is going to be represented by different colors. So for any given individual that we're kind of curious about, we see, okay, we know what their DNA looks like, and the lucky thing with us for most killer whales is we actually know who their mothers are as well. So we can say, okay, this individual got 1 pink star and 1 blue triangle from their mother, so they must have gotten a pink star and a green triangle from their father. So now we can go back to the population and say, okay, which one of you might have a pink star and a green triangle, and lucky bee, you are the father. However, when it comes to human paternity testing, women can be really helpful most of the time in narrowing down the number of possible mates. Now, killer whales aren't always so forthcoming, so we kind of have to consider all biologically possible fathers. So at the time of conception, any male that could have been old enough, so at sexual maturity, but a male that still was alive. So the male could have died before the animal was born, but had to have been alive kind of at maturity. So we have to consider all of these possibilities. Now, instead of just looking at kind of 2 shapes, we want to expand the number of microsatellites that we're looking with, because that's going to help us narrow it down, especially with so many potential fathers. So we're looking at 25 to 29 actually microsatellites. And we're doing this for actually 200 plus Bigg's killer whales right now. And so once we actually know who they're mating with, so once we can start ticking off, you are your father, this is your father, instead of doing these nice family reunions, we're actually going to start looking for patterns. So some work that DFO has done has started to look at and found this kind of inshore-offshore divide from some certain Bigg's killer whales. So now is this actually a genetic kind of 2 distinct groups, or are we looking at kind of a behavioral, I live inshore, I'm going to go offshore to mate? And that way they might be finding using this as a barrier of some sorts to prevent inbreeding. Is there kind of a north-south divide, or is there some kind of other nondescript traits that we're not picking up on? So whether is it some acoustic cues that we don't see, is it a behavioral trait that we're not really in tune with at this point that they might be using? Because we still see really high rates of diversity, we don't see really high levels of disease, so we know they're really good at avoiding inbreeding, but what triggers are they picking up on? And that's really what we're trying to look for. So in the meantime, I'm stuck in the lab doing a heck of a lot of paternity testing right now. And I really need to thank people that made this possible. So the Killer Whale Adoption Program and Mr. Doug Horswell provided a lot of the funding for this work to happen, and a lot of the sequencing work is actually being done here at the UBC's Genetic Data Center. So thanks.
Do you have any questions?
Me too. I think everyone, yeah, so the question is, everyone's so excited to know what I'm doing and what I'm finding, and that they want me to come back next year and tell you. So I will.
Comment on any of the paternity work done with the resident whales?
Yeah, so right now, there's actually a paper that's being prepped for submission, so I can't really speak much to it, but there is a paper that will be coming out soon on paternity work for southern residents. And as you know, there's really only one acoustic clan in southern residents, so with a small population, there might be higher rates of inbreeding inherently in that population anyway. So we're kind of a little bit kind of cautious about that, but some of our other work has done kind of looked at variation at important genetic sites and found that they have been able to retain a lot of variation, which makes it really healthy. So we've kind of got cautiously optimistic from a genetic perspective that there's kind of recovery potential.
We have a mic as well. We're going to try to see if we can get you on mic. Pass that down. Question. Oh, it's muted. It's not working.
I can hear you.
We thought we had it all worked out.
What samples you use to do the testing? What tissue or?
Yeah, so we have a really, the question was what samples we use to do this study, and we have a really large tissue collection that started, I'm going to say, in the early 90s, and Graham Ellis was a large part of that work. It was part of Lance's PhD work is when it started, and most of these are biopsy samples that have been continued with DFO kind of ongoing, but also with opportunistic carcass collections. So in this case, we're really interested in known individuals, and so with a lot of fresh carcasses, we can actually tell that as well.
Thank you very much, Carla.